Welcome to the Bac ID simulation. This simulation has two parts. In Part 1, you use the Catalate test to identify samples as being Staphylococcus or Streptococcus. In Part 2, you'll conduct 5 different tests to complete your bacteria identification of specific species of Streptococcus. To begin, select Part 1. Select and move the microscope slide to the middle of the tabletop. Select the marker and use the on-screen keyboard to label the slide. Select and move the 3% hydrogen peroxide bottle on the shelf to the slide. Select one of the 7 bacteria samples from the shelf and move the sample to the tabletop. Select and move the inoculating loop to the burner flame, then to the bacteria sample, and finally to the slide with the hydrogen peroxide. Select the magnifier and view the sample to see if bubbling occurs. Bubbling indicates the presence of catalase, which is found in Staphylococcus, but not Streptococcus bacteria. Repeat this entire process with the 6 other bacteria samples and the 3 unknown bacteria samples. Once the Streptococcus bacteria are identified, select the End Test button at the bottom of the screen and then the Part 2 button. Part 2 consists of 5 different tests for identifying the specific type of Streptococcus bacteria. To begin the blood agar test, select and move to the tabletop one of the known Streptococcus bacteria. Select and move the 0.85% saline solution to the tabletop. Sterilize the loop by selecting and moving the inoculating loop to the burner flame. Move the loop to the plate on the tabletop containing the bacteria, and after collection, move the loop to the 0.85% saline test tube on the tabletop. Select and move to the tabletop the blood agar plate. Select the marker and label the blood agar plate with the name of the bacteria. Select and move a swab to the 0.85 saline test tube and then move the swab to the blood agar plate. Move the blood agar plate to the incubator. When the incubation period is complete, select the magnifier and observe the plate for bacterial growth. Repeat this blood agar test for the other samples of Streptococcus bacteria. Do not test any Staphylococcus bacteria using this procedure. Select one of the unknown Streptococcus bacteria samples from the shelf and repeat the procedure to identify the unknown bacteria. To begin the bacitracine test, select Part 2 and then select and move one of the known Streptococcus bacteria to the tabletop. Prepare the saline test tube as you did previously. Make sure to sterilize the inoculating loop before transferring the bacteria from one plate to the saline test tube.